back and forth between T cells and B cells. So we've looked now at both B cell and T cell maturation, essentially in development. Now chapters 11 and 12 will again tick back between T cells and B cells in terms of activation, differentiation, and memory. So we'll do T cells in chapter 11. We'll do the same thing for B cells in chapter 12, and then 13 is sort of finally these kind of bring it all together chapter in that we will look at the effector functions of activated, fully differentiated T cells and B cells in terms of what are these antibodies exactly doing, what are the T cytotoxic cells exactly doing. So that'll be chapter 13. And then I think the final two chapters that we'll get to are 15 on allergies and 17. Yes, and chapter 17 on vaccines. I think that'll be the four chapters for the final exam. Okay, so let's turn our attention back to T cells after a little sojourn with B cells and look at once they've now matured and developed into formal mature T cells, what kind of happens next? So what's the next stage? So the next stage is they get activated by antigen, they will differentiate into specific T cell types, and we'll look at aspects of memory, T cell memory. Okay, so the sort of T cell activation story begins with the T cell, with what we'll call the naive T cell here, either a CD4 positive or a CD8 positive T cell, interacting with some form of antigen presenting cell. That's the sort of quintessential first step to activating a T cell. Very often these, the initial antigen presenting cell is going to be a dendritic cell, but there are other types. We'll look at that a little bit later in the chapter. And this antigen presenting cell itself has either phagocytized an extracellular pathogen, processed it, put those processed pieces of antigen into MHC class 2, and that can trigger, or that can activate a naive CD4 positive T cell, or it itself has been infected, say, with a virus, and therefore those intracellular peptides will be presented in a class 1 molecule and can activate a CD8, a naive CD8 positive T cell. So this is sort of the overview of what we're going to be kind of looking at in this chapter. So that interaction, well, that first interaction through the T cell receptor and the MHC molecule is the first signal. There will be other signals that are involved. So this other thing that they're drawing in here, this co-receptor section will be signal 2, and that will essentially cause an activation through the release of a small storm of cytokines that some people refer to as signal 3. This activation process will lead to clonal expansion, so that activated T cell will start dividing into multiple copies of itself, and eventually those cells will go down several possible differentiation pathways to either an effector cell of some kind, and we'll see that there are various types of CD4 positive T cells, or memory cells, and the same thing for the CD8 positive cells. They can become effector cells, mostly cytotoxic T cells, or memory cells. This particular chapter is going to focus mostly on the T helper cell part of the story up here, the cytotoxic T cell stuff. More information on that in chapter 13, with the effector CTL part of the story. So we'll mostly be focusing on this top part in this particular chapter. So this is kind of a nice overview picture for what we're going to do. Okay, so we kind of think back a couple of chapters to those mature T cells that have gone through selection, have gone out into the periphery, are sitting in lymph nodes or the spleen or whatever. Both the CD4 positive, let's go back to here, both the CD4 positive and the CD8 positive mature but naive cells, and naive meaning they haven't seen antigen yet. That's kind of what that phrase means, or that word means. They are essentially in the what's called the resting stage, or the G0 stage. Of the cell cycle. So remember, if you're actively in G1, then you're preparing to divide. So the G0 is that, G1S, G2M. G0 is sort of that outside the cell cycle stage. And so they're just sitting there. So it's one of the few times where a cell is kind of literally just sitting there. So it's not preparing, it's not in G1 preparing to divide, it's in G0, really waiting for antigen, waiting for activation. So these naive cells have not encountered antigen yet, they're just kind of sitting there waiting. Now that doesn't mean they're sitting there waiting always in one spot. Remember the idea that, because I think we talked about this a little bit in the last chapter with B cells, these naive T cells can recirculate, right? So they can move from the spleen back into circulation, hit that lymph node, go back into circulation, hit that lymph node, right? So they have the ability to recirculate in an effort, presumably, to capture antigen or be exposed to antigen a little bit more readily. They're sitting in G0, but they can recirculate. That is one of their major abilities. So as we see in this overview slide, the first step towards this activation is interaction via the T cell receptor and on the T cell, and the MHC molecule on the antigen presenting cell is the first major step towards activation of that, whichever kind of T cell is appropriate to do this. Now, this kind of small bit of interaction here is actually more complicated, so this is the more complicated view of this. The successful T cell antigen presenting cell interaction is mediated through not only T cell receptor recognizing the appropriate MHC molecule and the appropriate peptide that's sitting there, but if we're looking at a CD4 positive cell, then we have the CD4 molecule also playing a role in recognizing the class II molecule. If we're in a CD8 positive cell, then we have CD8 interacting with the class I MHC molecule. So remember, it's a combination of the T cell receptor and either CD4 or CD8, depending on what type of T cell we're talking about. But then we also have other important molecules. Well, of course, we have the signal transduction portion of the T cell receptor, right? This, the trimer of dimers that are encompassed by CD3 for the signal transduction piece, as well as CD4 or CD8. Another important stimulatory piece to this interaction is shown here with CD28 on the T cell and either CD80 or CD86 on the antigen presenting cell. We're going to see shortly in the next slide that interaction, which is sometimes referred to as the co-receptor, is signal 2 that is going to be necessary for full activation of that T cell. And all of these things, the T cell receptor complex, T cell receptor in CD3, CD4 or CD8 depending, and CD28 to collectively create this thing that they abbreviate here called the C-SMAC. Who makes these things up? Which is the central supramolecular activating complex. Right, so that's the central sort of repository or location for all of these different proteins. And part of this is mediated through that idea of lipid rafts that we were talking about a little bit, I think, in the last chapter as well. all of these molecules sort of drift towards each other into those specialized regions of the cell membrane called lipid rafts. Right? But as you can see, there are actually a few other important interactions here that are more adhesion molecules. So CB2 and LFA3 and LFA1 and ICAM1 are more adhesion types of molecules that sort of not cement together, but help keep the two adjacent cells attached together long enough so that all of these other ones that are involved much more in signal transduction can get the signals, transduced in the right way. So these more peripheral proteins, notice C-SMAC central versus P-SMAC peripheral supramolecular activating complex. That's peripheral. So you have the C-SMAC with all this, mostly the signal transduction stuff, and then the peripheral supramolecular activating complex is more adhesion type molecules. But luckily, most of the story is the same whether we're talking about a CD4 positive cell or a CD8 positive cell. The major difference is the kind of MHC molecule and whether you have CD4 or CD8. All the other molecules are the same, so that's kind of good. So this interaction is, fairly complicated and requires a number of different both signal transduction pieces and adhesion pieces to keep the cells together in the right conformation so that all these signals can get transduced. Okay. Because, as it turns out, it takes several signals, at least two, really three, in the real sense, in the practical sense, to get this T cell fully activated. And that's what's summarized in this figure. And so what people call signal 1 is the classic T cell receptor and the co-receptor CD4 or CD8 interacting with the MHC molecule with the peptide that T cell receptor is against. Signal 2 is the co-stimulatory interaction between, again, CD28 on the T cell and CD80 or CD86 on the antigen-presenting cell. So that's CD80 or 86. This is CD28. And so those are worth remembering. Okay, because we're just going to keep seeing them over and over again. Okay, so CD28 on the T cell, CD80 or 86 on the antigen-presenting cell. And I'll show you an experiment in a second that shows pretty clearly that to get stimulation or get activation of that T cell, you need both signals 1 and 2. Okay, if you don't have both of them, the T cell is not going to get activated. And actually, not just is it not going to get activated, it goes down the other way, it's going to get inactivated. I'll show you an experiment for that in a second. And then realistically, full activation in terms of going further down this row, starting clonal expansion, starting differentiation, is going to be controlled, probably not surprisingly, by the whole array of cytokines that the T cell will start secreting on its own, and it will be hearing, seeing, whatever analogy you like better, cytokines from that initial antigen-presenting cell and from other cells that are going to be in the vicinity of both the innate and adaptive systems that will help push these activated T cells down their differentiation pathways. So that is now being referred to as signal 3, but initial activation requires clearly 1 and 2. This signal 3 is a little bit further down the road. So for most of the time, we tend to focus on the stimulatory or the activating signals. Okay, so we've already seen now a couple of times. Now CD28 on the T cell and CD80 or 86 excuse me, on the antigen-presenting cell. And that is going to be involved in activation of those naive T cells. So that seems very, very clear. And again, I'll show you an experiment or two on that in a second. But there are also negative ones as well that we'll get to in a second. Might as well cover the other stimulatory one first. The second one called ICOS, and then on the T cell, and ICOS ligand on the antigen presenting cell. And as you can see here, the ICOS is expressed on more types of antigen presenting cell than 1886. 1886 are mostly on dendritic cells. Okay, and we think it has something to do with not the initial activation, but maintaining that activated state. So the 2880 interaction is the first one that gets the T cell activated. Once that has happened, that activation needs to be maintained, and that seems to be the role of this ICOS and ICOS ligand interaction. So most people tend, and most of the time, textbooks tend to focus on the stimulatory, the activation part of the story, but there are also negative co-stimulatory signals available. Okay. CTLA4, PD1, and BTLA are all negative co-stimulators, which are thought, generally speaking, to start slowing down the process and turning it off as the pathogen is being cleared. So we've talked about that a little bit as well in terms of like T regulatory cells and things like that. So it seems that some of that negative co-stimulation is mediated by these particular proteins. And in a lot of cases, they figure some of this out through knockout mice. So they've knocked out these particular genes in a strain of mice and then see what is wrong with the mice. And those mice that have these genes knocked out tend not to be able to get rid of all the T cells that get activated on encounter with antigen. So they're less able to turn off the system, turn off the immune response at the end. So that leads people to believe that these have obviously negative kinds of regulation. So you can see regulation of T regulatory cells. And even during the process, during the actual immune response, you need to kind of keep those T cells somewhat reined in because otherwise, theoretically, they're just going to keep dividing. Okay? And that's not always a good idea. So you want to keep that under some kind of control, and that seems to be part of what CTLA4 especially is all about. It kind of puts the brakes on, uncontrolled proliferation. You don't want that, of course. So that seems to be, especially what CTLA4 is all about. Whereas these latter two seem to be more involved in T regulatory cells. But as usual, a lot of work is still being done on this. Notice the question mark there. So we certainly don't know the entire story for all of this. Okay, but those are some of the proteins you probably want to be at least somewhat aware of. And this is the experiment that shows that at least the first two signals are absolutely necessary to get stimulation of that T cell. So what they're showing here are just a couple of different situations where the T cell does not get properly activated. So for example, if you have this antigen presenting cell and you fix it, you throw glutaraldehyde or formaldehyde at it, before it has expressed any CD80 or CD86. So the only signal that the T cell is getting is signal 1, and it doesn't get signal 2. Not only does that not activate that T cell, but it creates a state that I think we've seen this term before, in case we haven't. That state is called anergy. So it's not that it just doesn't get activated, it gets inactivated. Because signal 1 by itself, as you see here, triggers not activation genes, but anergic genes. So it turns on a cascade that actually inactivates that cell. And if later you try to trigger that cell with signal 2, it won't work. So once a cell has become anergic, you kind of can't get it back, or at least not the traditional way, where now we have signal 1 and signal 2, but the cell has already become anergic and it's not going to give you a response. I think the context that we had seen this earlier was this is one possibility for what happens to some autoreactive cells. They don't get destroyed directly by apoptosis, but they become anergic. This is the same sort of idea. So if the T cell doesn't get signal 1 and signal 2 somewhat, together, it doesn't get, again, not only does it not get activated, it becomes anergic. And then this third experiment is sort of similar idea, but in this case, instead of killing the APC, it's a normal APC, so it gets signal 1, but you have an antibody that blocks the interaction between CD28 and CD80. So again, it's getting signal 1, but not signal 2. Same thing happens, that T cell becomes anergic. So it seems really, really clear that both signal 1 and signal 2 pretty much simultaneously are required for proper activation of that T cell. And that, again, it's not as simple as if you don't get them both, you don't get activated. It's actually more active than that, because it's an active process that makes that T cell anergic. So when you don't get both signals, that T cell is essentially taken out of circulation in a sense. It can't be reactivated. So 2 signals, pretty much the case. And then, like I said, most people now are recognizing these subsequent cytokine interactions as signal 3. Not necessary for initial interaction, or initial activation, but these cytokines will again sort of continue this process, resulting in clonal expansion and eventually differentiation of the cells. But people are starting to use the term signal 3 for those cytokines. So we're kind of focused on the first part of this with these antigen-presenting cells. So figure 5 is kind of a nice summary of the 3 major types of antigen-presenting cells. The major one being the dendritic cells that can clearly activate naive T cells. The other 2, macrophages and B cells, it's kind of unclear whether they can activate naive B cells, but they can certainly and very clearly activate ones, or continue to activate effector cells and memory cells. But dendritic cells seem to be the only ones that are very, very good, at least, at activating naive T cells, ones that have never seen antigen before. So the rest of the table just kind of compares the 3 types. A lot of more similarities, I would say, than differences among them. So they can all take up antigen from outside the cell. Okay? They can, well, there's a little bit of difference in terms of activation of the cell itself. The dendritic cell can be activated by those pattern recognition receptors. So there again is the connection to the innate immune system, as well as macrophages. B cells, and we'll see this more in the next chapter, B cells are mostly mediated by antigen recognition through the antibody that's sitting on their surface. Okay, so B cells are clearly not really part of the innate system, although having said that, there are of course always exceptions. There's a subset, a subpopulation of B cell that looks very, very much like an innate cell, so we'll take a look at that in the next chapter. Since especially for exogenous antigen, we're talking class 2 molecules, so as the antigen presenting cell itself is activated, it's going to increase expression of class 2 molecules, which makes sense, because that's how it's going to activate the T cell. They all use CD80 or CD86 as the co-stimulatory ligand. That'll hook up with CD28 on the surface of the T cell. I've already talked about the kind of T cells. And then one slight difference among these types of antigen-presenting cells is where they typically are. Okay, so they can all circulate. Dendritic cells and macrophages can also be found in the peripheral tissues, meaning pretty much everywhere, okay, because they're also really part of the innate system, so they can go anywhere, whereas B cells are going to be in the follicles of lymph nodes in the spleen. Again, more on that in the next chapter. And then once they're activated, they can all pretty much, well, actually, never mind. The dendritic cells can move towards either the spleen or the lymph nodes in order to activate those T cells, but they can also stay in what's called tertiary tissues, which is, I don't know why they use that term actually, but that tertiary tissue is actually the site of infection. So they can stay there and possibly activate effector cells and memory cells that are also in those locations. Macrophages and B cells are either going to move to or stay in the lymph nodes in the spleen to activate T cells in those locations. More on that in a little bit. So there's a lot of detail here that you don't need to worry about too much, but most of it's really kind of the same for all of them. There are only slight differences among the three major types. And then the final part of activation is sort of the weird situation of superantigens. So there are antigens and there are superantigens. Superantigens are really scary because they have the ability to activate T cells non-specifically, which makes them really kind of frightening molecules. So as you see here, they have the ability to sort of cross-link a antigen-presenting cell and a T cell, in this case a T helper cell, because they have the ability to interact with specific beta chains of certain T cell receptors. So that's sort of their claim to fame. So they sort of clamp onto beta chain of the T cell receptor and hook it up to an antigen-presenting cell. So this T cell receptor doesn't really recognize whatever that peptide is. That could be a self-peptide, it could be anything at all. It doesn't matter. But this acts as sort of a vise or a clamp that connects the two cells together completely inappropriately, completely non-specifically. The only specific part of this is that certain superantigens can do this with certain beta chains. And that's shown in the next table. People have worked out for specific superantigens which beta chain of the T cell receptor they work through. So that's what all these numbers are, which of course you don't need to memorize. But they mediate certain disorders like food poisoning and toxic shock syndrome, I think was the first one that this was kind of worked out for. Scarlet skin syndrome, if you ever come across that. And some people are linking it to things like autoimmune disorders like arthritis and just shock in general, but rheumatic fever seem to all work through specific, I think all of these are bacterial, right? Yeah, all these are bacterial, but there are some viral superantigens out there as well, but most of them seem to be bacterial. So this can be any, this blue thing here representing the superantigen is, any of these things, so staphylococcal enterotoxins, exfoliative toxin, whatever. So if it's, say, toxic shock syndrome toxin, and you have T cells floating around with beta chain number 2, then all of those T cells could be activated by that particular toxin, no matter whether or not, and usually not, the T helper cell actually is against that particular peptide. So it creates this enormous activation inappropriately of these T helper cells. This is all a class 2 problem, right? Because it's alpha beta. So it's all the T helper cell. So it makes this huge proliferation of T helper cells inappropriately, which can then result in just this broad shock kind of to your system as your immune system is turned on radically. It's not just on; it's activating all these different T cells, which is really, really a problem because each one of these numbers can be up to about 5% of all of your T cells that are getting activated non-specifically. Okay, so they're all getting activated, they're all starting to proliferate, they're all starting to churn out all of these cytokines that are causing inflammation and vasodilation and all those other kinds of things that collectively, on this enormous scale, we recognize as sort of shock, quotation marks. Okay? Shock can be deadly, right? As your blood pressure goes down, your heart starts racing, your core body temperature goes out of whack. All of those in combination can be lethal. So this is kind of a big deal and quite dangerous for these specific toxins, and the list keeps growing as we understand kind of more about this.
How do the superenergies come about?
They are proteins produced by these bacteria. Yeah. So these are just proteins encoded by these. By these bacteria. Okay, so this is how staph causes food poisoning. Okay, so these are the toxins. Now we didn't know exactly how they worked, but now we know that it's not just the traditional toxins. Let me be clear. Not all bacterial toxins are superantigens. Okay, the traditional toxin just affects the cells in a much more direct way, and has nothing to do with the immune system. But this small group are called superantigens because they have this ability to involve the immune system, and therefore the result is much, much more dramatic and also kind of much more lethal. So staphylococcal food poisoning is worse than your garden variety form of food poisoning, because you have all of these T helper cells going out of control. So the book does make one little distinction, and it's a minor one, but since it's in the figure here, I'll point it out. There's called exogenous and endogenous superantigens, and the only difference is it completely soluble, or is it attached to the cell? That's really the only difference. So an exogenous superantigen is one that's completely secreted / soluble. An endogenous superantigen, you get the idea there. The dotted line there is where the antigen, or the toxin is still connected to the cell. So if it's just the secreted protein, that's an exogenous superantigen. If that's connected to the cell, the infected cell, then it's called an endogenous superantigen. It's not a big deal, but they do make that distinction. And I honestly don't know which ones are which here, so it's nothing you have to really worry about the terms that are out there. Okay, so that's a little bit about activation, both appropriate and inappropriate activation of these T cells. So as we've been seeing here and there, any time you kind of inappropriately activate these things, you're asking for trouble. Okay, I think that's where we're going to stop for today. The other half of the chapter is going to deal with differentiation and memory. But I can't get that done in 20 minutes, or actually in 10 minutes.